inspired your research topic, and why do you think it's important to investigate?
Autism spectrum disorders represent 1% of the general population, approximately. And for 80% of those patients, there is no genetic cause known. In our work, we decided to work on syndromic autisms, and autisms that we know the genetic cause, so we created a model of cell lines with that mutations to investigate how those cells becoming neurons are developing and how their gene expression profiles is presented. That way, we'd like to see if from one syndrome we're able of building the link between the cause, the cell development, and the patient conditions. We expect to find many molecules that could lead us to treatment.
How do your findings contribute to the existing body of knowledge in your field?
So in this work, we study essentially the effect of the knockout in FMR1 gene. And this was important because it allowed us to have a model with a non-gene that is altered. But what we observe when we look at the expression of different genes and in light into the function of the neurons and how active they can be, we realize that there are common aspects when we look at the big umbrella of autism and we call it functional convergence. We do observe there are common phenomenon and common signaling pathways that are altered in a process. So this was a very interesting finding.
What future research directions do you envision based on the outcomes of this study?
So our studies has shown that cells are affected in many ways by the mutations we were looking at. So we would like to see for each cell how the developmental program is affected.
And the other thing that I'm currently developing is for sure to work on 3D models. And this is very important. In our paper, we worked essentially with 2D cultures with neurons, but we know that our brains are composed by many cells and crosstalk between different cellular types that give support to the neurons are important. And we do want to look at this crosstalk and this connectivity between the different cell types in the brain in different genetic backgrounds if you can. I think it is the way to develop the field.
Thank you for watching, and check out the link in the description for the full paper.